This video will be the second part of Chapter 16, Control of Gene Expression in Bacteria. We'll look at transcriptional attenuation, which is a form of transcriptional control, and then we'll also look at several forms of post-transcriptional control in bacteria. So the tryptophan operon is the example we're going to use as we look at this example of attenuation. So tryptophan can be made by bacterial cells, that's an amino acid. In order to make that amino acid, there are 5 genes that are needed. And so these 5 genes are the trpis, D, C, B, and A gene. And this is actually just showing the synthesis of that amino acid. And each of these steps are catalyzed by these enzymes that are encoded in the trp operon. These 5 genes all share a common promoter and regulatory system. For that bacteria, it would be best to turn on that operon when tryptophan levels are low, so you can make your own tryptophan, or if you already have tryptophan from the environment, you don't need to transcribe that operon and you would turn it off. So there are actually 2 ways that the tryptophan operon is regulated. First of all, it has the standard repressor, very similar to the lac repressor that we talked about before. Second is a mechanism called attenuation, and this is what I'd like to go into now. So there is a 5' UTR in this operon right before you get to the first gene, which is the trpE gene. This region is very important in the regulation of the transcription of the rest of that operon. This is kind of complicated, but I like to point out that there is actually a region that codes for a protein. So this red region here, region 1, codes a very small protein, 14 amino acids, and importantly, 2 of those amino acids are tryptophans. And it turns out that the translation of that very short protein regulates whether the rest of that operon is transcribed. Now, this is a little confusing because we're talking about translation regulating transcription, which should theoretically happen earlier. Remember in prokaryotes that we don't have a separate compartment for transcription and translation. And so as soon as you start translating a messenger RNA, it will get bound by a ribosome and start to be translated. So when that ribosome gets to those tryptophan codons, either there's going to be low tryptophan levels in the cell and the ribosome will pause because it can't find a charged tryptophan tRNA, or there's high levels of tryptophan in the cell and it will just go right on through there without pausing at all. Whether the ribosome pauses or doesn't pause is going to determine whether the rest of that operon is transcribed or not. Now, the way this works is actually due to secondary structure of the messenger RNA. And so here we have detailed these 4 regions of the messenger RNA in that 5' UTR. And there are different ways that those regions can base pair together. Now, this is the 5' end of that messenger RNA, and as we go ahead and make the RNA, it will start making these hairpin loops with the RNA base pairing with each other. So there are 2 possible ways that that RNA can form secondary structure. The first way is that if that 2 region is not available to pair with the 3 region, the 3 region is actually going to pair with the 4 region. If the 3 and 4 region base pair with each other, that actually forms a transcriptional terminator. So here is that intrinsic transcriptional terminator we talked about previously. You have that hairpin loop followed by a string of U's. So if this particular structure forms, you're going to stop transcribing that operon. In contrast, if the 2 region base pairs with a 3 region, the 3 region is prevented from forming that hairpin loop with the 4 region, and therefore it will not terminate and will continue transcribing the whole operon. Now, what really determines which of those secondary structures form? So the ribosome is what determines it. So when there is high tryptophan in the cell, that ribosome will bind onto that small leader peptide and start to translate it. And it will go sailing right on through that region with the tryptophan codons because there's plenty of tryptophan tRNAs in the cell and therefore has no trouble finding it. If this happens, the result is that this ribosome is already into that 2 region by the time the 4 region is transcribed. And this will prevent the 2 region from base pairing with the 3 region, and therefore the 3 region and the 4 region can base pair together. This will form that terminator, that transcriptional terminator, and we will stop transcription. In contrast, if that ribosome stalls when it gets to the tryptophan messenger RNAs in that little protein, it can't find the tryptophan tRNAs because the levels of tryptophan in the cell are too low, then it's going to sit there on that 1 region. As a result, you continue transcribing and the 2 region will be formed, next the 3 region will be formed, and as soon as those 2 regions are transcribed, that RNA is going to base pair together. So the 2 region base pairs with the 3 region and we prevent the formation of that transcriptional terminator, and therefore we go on to transcribe the entire operon. I just want to point out one way to think about this is, what does the cell want to do? So if tryptophan levels are low in the cell, it needs to make those proteins that are encoded in the trip operon so it can synthesize its own tryptophan. And so the result is you want to keep transcribing if the ribosome is stalling. On the other hand, if the ribosome has plenty of tRNA, you would prefer not to waste the energy of that bacterium by going ahead and making all those proteins if they're not really needed. Okay, the second part of this video will be on post-transcriptional regulation. Transcription occurs, you have the messenger RNA, and the regulation is really at the step of the RNA. So between this step, RNA to protein, there's some type of regulation. We're going to talk about 3 different examples of this. So antisense RNA, something called riboswitches, and also ribozymes. So this first example, antisense RNA, I'm going to tell you about this gene called the OMP-F gene. So this encodes a protein that makes a channel for passive diffusion across the membrane. If the bacterium gets into an environment with very high osmolarity, it's going to actually suck all the water out of that bacterium, and you've really got to close off those passive diffusion channels very rapidly. So in order to do this, there's a couple of possible approaches. One way would just be to turn off that OMP-F gene so it's not transcribed anymore. But that is not going to give a really fast response. So there's still a lot of existing RNA and protein that's in the cell, and that cell might die, in fact, before it gets rid of all of it. So what the cell does is it turns on this functional RNA gene, it's called MIC-F, and this gene is actually complementary to the messenger RNA for the OMP-F gene, called an antisense RNA. And it binds to that messenger RNA and it prevents it from being translated. Riboswitches are another kind of regulation of RNA. So this is actually done by a regulatory protein. So this protein will bind to a messenger RNA and make that messenger RNA form into a particular secondary structure. When the structure is present, the ribosome binding site is actually hidden. So therefore that messenger RNA will not get bound by ribosomes and the gene will not be translated. In the absence of that regulatory protein, that messenger RNA forms a different secondary structure. Then that ribosome binding site, the Shine-Dalgarno sequence, would be accessible and the ribosome can bind on there and translate the protein. The last example is something called a ribozyme. So a ribozyme is actually a RNA that has catalytic activity on its own. And what happens in this particular gene is there are 2 secondary structures that the messenger RNA could take. And one of those causes it to actually cleave itself and break itself down. So this particular gene encodes this RNA. You make this protein, glutamine 6-phosphate amidinotransferase, and the product of that protein is actually a molecule shown in blue here. And this product will bind to the messenger RNA that encodes that enzyme, and when that particular effector is present, that changes the conformation of the RNA, and the RNA degrades itself.